in office right after class till about 1, and I'm eating over at Spader. And I'm coming back, I'd be there till 3:30, fly under the door, that's,
Yeah.
All right.
Okay.
After that, I'll be there.
All right.
Okay, I suppose we should get started. So those who've won it, review session after lecture today. If you've got questions about the assignment or what we're looking for on questions, that might be a good place to check those. We'll do a break. Middle, we got assignment 2 due now. If you want to pass those, they're kind of a common area, either forward or collect them as a group. Yes, I do up here. First question, due by a week from today. If you ask questions during class, write down that question and that counts. That could be procedural or content, doesn't matter. And then I believe I've got assignment 3 posted. You should have gotten a hard copy with the handouts just now. So, assignments. You got them? Anybody else? Okay, so last time we talked about nucleic acids, and we started to put together the connection between DNA and protein. The ability to go from DNA to RNA, transcription, making a copy. Going from RNA to protein, we're translating from a nucleic acid language, using those codons, those 3 nucleotides in a row. Each one of those codes for a specific amino acid. With those amino acids, we can form a protein that'll fold into a shape that serves its function. We put those nucleic acids into DNA, and we talked about why the rungs are always constant. So you got 3 rings going across, and then you match up the number of connections, the hydrogen bonds going across. So that way, A always matches with an A, G always, A matches with a T, G matches with a C, and that copies, that code is conserved. We need that to be more or less constant all the time. So transcription and translation, making a copy, that copy leaves the library, which is the nucleus, goes out to the protein synthesis machinery, the ribosomes, And there we have translation out in the cytoplasm of the cell. RNA is converted to protein. Hopefully translation. The rubric or the Rosetta Stone for this is this chart that I gave you last time. The codons with the 3 nucleic acids in a row all code for specific amino acids that are used to build those proteins. Sometimes we need all 3 to get to a specific amino acid. Sometimes we just need the first 2. So that third spot over here for threonine, doesn't matter which nucleic acid is there, we're still going to get threonine. And that happens in a number of different cases up there. So we left off last time talking about how we interpret the genetic components or the genetic worth of an animal. So has anybody in your Dairy Quiz Bowl days genotyped? Equals temporary environmental effects plus permanent environmental effects equals phenotype. Has anybody heard that before? Genotype is what the genetics are. Basically, the codons that are there to code for protein, the genes that are present, the nucleic acids in a certain order. Permanent environmental effects might be an animal gets sick as a calf, damages its lungs, or has lameness. Something in its pattern of life happens to the animal and it sticks with that animal and influences the performance of that animal. Temporary environmental effects might be the weather today or the feed they get this week versus next week. That'll influence how she expresses or how she performs, our phenotype. So in those first 3 instances, we're focusing on the phenotype. And then through mathematics, we can say, okay, all those ancestors have this in common, therefore this animal probably has it as well. We're going to say that's a genetic component. What we're taking is working with pedigrees, working performance of the animal, working with the progeny of that animal, taking our phenotypes, and then mathematically with large numbers of animals, except for the performance one, inferring what the genotype is. Because in theory, all that can be passed from generation to generation is the DNA. Now, there are some arguments to that. There are some things that suggest that the performance of an animal here might affect the permanent environmental effects might go through the egg in some way, shape, or form, but we'll see how that goes through. With genomics, we're looking directly at the DNA. Now, what's going to happen in your lifetimes is we're going to get better at interpreting what we see. Well, there's a guanine there. That means she's going to be tall. We might be a little bit shaky on that one. We're looking at DNA directly in a number of different spots. So the bovine genome, 3 big gigabytes or 3 billion pairings of A and T and G and C. Put it in a specific order, get you a cow. Those pairings are divided into 22,000 genes, 14,000 of which are all common to mammals. So there's a repetitiveness of the genes that occur. Bovines share 80% of their genes with humans. Most of the metabolism work and how things work was figured out on bacteria. Krebs citric acid cycle. Cows are less similar to humans than rodents. Humans and rodents belong to a group called the super primates. There's also about 1,000 genes that you're going to find in dogs and rodents that are not identified in humans. So if we lay out those 3 billion nucleic acids in a row, our plan is to take certain spots and look at them and see if there's variation in those spots. Is there an A there? Is there a G there, a C, a T there? And does that correlate to something we're seeing with that animal? So SNPs, has everybody heard of SNP before? A SNP is basically the basis of genomics. That there are spots where there is variation in the genome in these animals. You got 3 billion nucleic acids lined up in a row. There's going to be spots where we have variation, and those spots are named single nucleotide polymorphism. In that 2,568 spot, there's variability for some reason. So tracking the variation at the nucleotide level, most of the bovine genome is the same across all cows. 99.999 plus nucleotides are the same for all the animals. Why do you think that might be? What if there was only the same in, say, 97% of the spots? It would be a banana. They're cows, they're the same, they have to have most of the same stuff. But there are spots along that 3 billion base pairs where things seem to vary. And the question is, does that variation lead to some phenotype difference, lead to some performance difference? Maybe a little different amino acid in a protein. Maybe things are a little stickier there for RNA or something. So I think the last number I found, they're looking at 40,000 out of 3 billion that seem to have differences. So you go through, you look at those 40,000 spots, you note for this animal which of the 4 nucleic acids are there. And then you say, okay, this cow has that pattern. Most genomic tests don't go near that high. I think we're up to a couple thousand spots. Where have I seen that pattern before? This cow's got an A here, a T here, a G here, and CC here. Well, last time I saw that, that cow was a very nice type cow, or had very high production. So given what we see in those individual spots, we correlate it back to the information we have in the database, and we say, yes, this animal is going to be a strong performer in the show ring. This animal is going to be a big producer. This one's going to have calving ease. So we're just in the infancy of all that. As time goes by and our database builds, we're going to have better understanding of what that individual variance may include. So how does the variability or SNP make a cow better or worse? What might happen? Different nucleic acid might lead to a different codon, a different amino acid, slightly different protein. That protein works better or less than another animal's. Or we have some sort of stickiness with the starts and stops, something that will influence how the protein is made or processed or something like that. So effectively what a SNP is a mutation. Variability in that one spot is a mutation across the animal. They are not lethal mutations. They are not mutations that are going to cease for that animal to be a cow anymore. But we have differences there that might give the cow or calf a competitive advantage in some arena, some level of production, some set to the hock, something. So those genes are going to express proteins. Maybe those proteins do the job just a little bit better. We get a little bit more milk out of that cow. Or we have a little bit more digestive enzymes. Things get absorbed better. Or she's a little taller. Something. So genomics, they mark those SNPs, those places where things are different across a range of cattle. And then you look for patterns. Which animals are higher type? Which animals have higher production? What pattern of variation fits those? So there are correlations, and you're looking at multiple spots. Loci means spots. For quantitative traits. Multiple genes are going to interact on some level and give us a phenotype at the end of it all. So we're looking for correlations. So what are the advantages of genomics? Has anybody worked through this before? It's a direct look at the DNA, which is what we were trying to interpret with the others. So when is the DNA set for an animal? Sperm meets egg, DNA is set. As soon as I can pull a gene, set of genes, that doesn't kill the animal, I can run a genomic test. So I can't do it at the one cell level. If I pull the nucleus out of that cell, everything's dead. But we may be doing this in utero soon. Certainly, as soon as the animal's born, we pull those hair follicles or clip a piece of skin, however it is, to get those genetics. It's a direct look. What's the downside of genomics? Only as good as our correlation models are. Starting out, they're a little shaky, but in your lifetime, I'm guessing they're going to be pretty robust. You might get up to doing all 40,000 if it gets cheap enough. Looking at all those differences and how they relate to a trait or performance that we're looking for. So you can do it early, you could do it fast, it's a direct look, but it's only as good as our interpretation of those SNPs. Anybody know how long it takes to work through your pedigrees? Probably something we can work on as soon as we choose that mating. But there's a lot of randomness in that formation of gametes, the sperm and the egg, that we have trouble accounting for in one individual animal. So performance testing or getting through and figuring out how much milk is a bull, is in a bull, how long does that take? We do traditional progeny testing. At least 4 years. Collect that semen, you get pregnancies, have a group of calves about a year later. It takes 2 years for those animals to grow up, a year to have performance testing and or get some sort of classification done on them. So you collected that bull, maybe at 15 months, maybe a year. And that animal's got to sit for 4 years until the USDA figures out through performance testing whether that bull is worth having around. Costs a lot of money to keep those bulls around for a long period of time. Whereas as soon as the animal's born, I send off a test. If I can get the same results and not have to wait 5 years, a year to grow up, a year to gestate, 2 years for those animals to grow up, a year for performance, the bull studs are very interested in saving that money. So there's an online video back from the 1960s. I'm not sure I can show it here and keep this recording going, so I'll give it a try, but
together of amino acids to form a protein. We know now that the three-dimensional structure and the function of a protein is determined. Only rarely is there an opportunity to participate in a molecular happening. You're going to have that opportunity, for this film attempts to portray symbolically, yet in a dynamic and joyful way, one of nature's fundamental processes, the linking together of amino acids to form a protein. We know now that the three-dimensional structure and the function of a protein is determined by the order of amino acids along the backbone of the molecule. So protein synthesis involves programming and assembly. In this film, the people portraying molecules, using the DANS idiom, tries to animate these two processes, the programming and assembly of a protein. Our genes carry the instructions for ordering the amino acids of each protein. Those instructions are encoded in a messenger molecule, an RNA, depicted in this film as a long snaking chain. Each of the message units is played by 3 adjacent people in the chain. Colored head balloons indicate the bases: green for guanine, blue for uracil, yellow for adenine. If there is a message, there must be a way to translate that message. That's the job of the ribosome and of the transfer RNAs. The ribosome is composed of a large and of a small subunit, and these are depicted in the film as tumbling, rolling clusters of body, amorphous by themselves, but organized and structured when in the act of translating the message. First, A small subunit, with the aid of an initiation factor, captures the message. Then, the first transfer RNA, carrying its cognate amino acid, is brought to the ribosome message complex by a second dancing initiation factor. This requires energy, and that's represented by a puff of smoke. Next, the large ribosomal subunit tumbles into place, and then the process of bringing each amino acid to the ribosome message complex is accomplished by the T factor and its GTP cohort. And so, in the order prescribed by the balloon colors in the message chain, each amino acid is brought to the assembly site to be added to the growing chain by the peptidyl synthetase. Next, in an energy-requiring step, the message RNA, tRNA complex, is shifted so as to bring the next message unit into the ribosome to allow the process to repeat itself. At the end, the terminator factor, seeing the termination signal, cleaves the completed protein from the last tRNA, releasing it from the ribosome. So that the ribosome can do its job again, the two subunits are split apart and separated from the messenger by the ribosome releasing factor. My diagram is of necessity static, but protein synthesis is a dynamic process. This movie tries to bring those dynamic interactions to life.
Was brillig and sly the 30S ribosome did gyre and gimble in the wave. 30S ribosome. All mimsy was mRNA, that colored message unit array. Found their work in the glorious sun by initiator factor 1. Initiator factor 2 went searching for tRNA, who bore the flaccid amino acid. Then, as the uppish bonds were formed, the 50S ribosome untamed came whiffling through the tallgy grass and burbled as it came. 50S ribosome! 50S ribosome! C factor plus GTP gathered in tRNA. Gathered in tRNA and came galumphing back. Amino acids both inside, alas, the peptide bond was formed. And as the next step took place, the message units shift a space.
So, a little bit different, but if nothing else, it shows that there's lots of things up on the internet that can help you understand transcription and translation. So I think there were some additional substances involved in this exercise, one way or another, but everything there is pretty much what we've talked about: transcription, translation. All those things are happening. Everything's matching up the way it's supposed to. Peptide bonds are forming. And there are a lot of other, probably less, what would you call it, hallucinogenic videos available on the YouTube to look at the formation of DNA from proteins. So what are your thoughts related to that? Completely ridiculous? I still have a student that comes up to me and every time he sees me and goes, tRNA, very loudly. So from this video, so I saw him at a wedding this past December. So lots of different ways to learn things. So get out your letters. Let's make sure we got our vocabulary down. Medial versus lateral. Medial versus lateral is what? A, left and right, B, top and bottom, C, front and back, D, center and side. Probably the best answer up there is D. Well, we could sort of see why you went with left and right. The medial plane is certainly in the middle, that splits left and right. But medial versus lateral closer to the center versus closer to the side. Superior versus inferior. Same choices. Probably depends on the creature you're looking for, either B or C. So B would be more appropriate for what? Human. Front and back would probably be more appropriate for our cows. Cranial versus caudal. So I'd probably lean towards C. But again, if you had humans, it might be B. Posterior versus anterior. Lean toward probably C. Antenna, anterior, posterior, butt, so front and back. Dorsal versus ventral. B is probably the best answer there. Amino acid sequence of a protein molecule is called its what? Primary, secondary, tertiary, quaternary structure. Amino acid sequence is primary structure, A. Final shape of a protein molecule is called its what? Primary, secondary, tertiary, or quaternary structure. Tertiary is the final shape of an individual protein molecule. How do we get quaternary? When two or more different proteins come together. Final shape of a protein molecule is due mostly to what? Coils and sheets, the sequence of amino acids, the R groups interacting with the water. Never force anything, get a bigger hammer. So the interaction with the R groups with the water is probably the best answer up there, but B and C are equally correct in the sense that the sequence of amino acids will determine where the R groups are and how things will shape and turn and twist. Any questions on any of that? Last set of compounds we're going to look at is lipids. Formal name for a group of that we commonly call fats. Lipid is a formal name for a group of compounds that are insoluble in water, which makes them hydrophobic. Hydrophobic. So they do not like water. On some level, they're going to run from it. So that makes them hydrophobic. Hydrophobic lipids are also non-polar, except for maybe the very ends. So non-polar, mostly hydrocarbons, with a little oxygen here and there. They're opposite of water in that way. They don't like being around water. They like to be by themselves. So if we have a nonpolar molecule, what it's made up with compositionally should be no real surprise at this moment. Mostly hydrogen and carbon. We look at our big four chart. Lipids are going to have carbon and hydrogen and a little bit of oxygen here and there. Nothing else really. So if we look at our chart, main thing we're looking for, hydrogen and carbon with our lipids, and we're going to have some oxygen here and there. Find the charts. So on the big four chart, no nitrogen, a little bit of oxygen, yes to hydrogen, yes to carbon. So one of the things when we're dealing with lipids one of the important properties is whether they're saturated or unsaturated. Does anybody know what we mean when we say that? Saturated versus unsaturated.
Completely filled and then not filled?
Completely filled with what?
And then not filled.
Not filled? So the question is, what is that? That's hydrogens. Completely filled with hydrogens or not. Probably the simplest way to put it. So does a lipid have as many hydrogens as it could possibly have? If it does, attached to that backbone of carbons, then we call it saturated. So hydrogen bonds as it can, sort of the same, saying the same thing there. So if it does, if the answer is yes to both these questions, we call it saturated. If not, we call it unsaturated. So saturation, if a lipid molecule is saturated, that molecule, that compound, has as many hydrogens as can be attached to it. Everybody sign in the attendance sheet. If it's unsaturated, we've lost some hydrogens somewhere along the way, and there's room for more. If those hydrogens are taken away, we have available bonds on our carbons. And if they're adjacent carbons, we'll go, well, let's form a double bond. So instead of just one connection between the carbons, we have two. And that influences a number of different things physically. We may have a situation where there's one double bond, we're missing two hydrogens. Or we may have a situation where we're missing a number of hydrogens that'll lead to more than one double bond. 3 and 6 places where we could have two more hydrogens, but we don't. So what are we looking at conceptually, from a drawing perspective? So we have a backbone on our lipid, chugging along, carbon, 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 carbon. We have probably two hydrogens stuck to it. It's making bonds with 4 different atoms. If we have an unsaturated situation, we have a double bond between the adjacent carbons. They're bonding there twice. And one of the important things to note is once you do that, that spot in the molecule can't spin anymore. The previous, we can spin around that. There's only one connection we can twist. Double bond, you're restricted. The movement can't happen anymore. You can't twist around that double bond. You have now a rigid spot in the molecule. So we probably have a carbon here, a hydrogen here, hydrogen here, but you've got that double connection there. We've lost 2 hydrogens, and the carbons laid back, well, we'll form bonds with each other. We don't need the hydrogens. They went wherever they're going to go. We'll just form a double bond. What the hell? So when we're thinking about saturated and unsaturated, it influences structure and form in the body and ultimately the purposes of those lipids. Talking about types of lipids in the body. These are the ones we'll cover in this class. Triglycerides is our primary energy store. We'll go through these one at a time. Phospholipids, they make up our membranes. Steroids do a number of different things. Their building block is cholesterol. Talk about how that happens. And then there are waxes, usually associated with outer surfaces and places where we're trying not to dry out. So like I said, we'll go through these one at a time. So triglycerides. So with our fats, we don't have polymers like we had for our carbohydrates or proteins or DNA, but we do have still those connections that involve a dehydration reaction. A triglyceride is made of one glycerol molecule, which is a sugar, and it's got 3 carbons at its backbone. So you've got 3 carbon sugar, open form, not a ring form. And you're going to have, hanging off of that, three fatty acids. So these squiggly lines here represent anywhere from 4 carbons in a row to 36, 40 carbons in a row. And we have a connection here that's a dehydration reaction between the two. So a glycerol molecule with 3 fatty acids hanging off it. So here's a drawing, what that looks like. We've got your glyceride molecule right here. Dehydration reaction connects these 3 fatty acids to that triglyceride. The fatty acids vary in length. How long are they? They vary in the number of double bonds that are there. So if we're looking chemically up here, we have 2 saturated fatty acids varying in length. Right here, we have an unsaturated because we have 1 double bond in there. So that would be a mono unsaturated fatty acid. So the purpose of triglycerides is to store energy for the animal for future use. Very energy dense. We take that small amount of energy on a gram basis. There's more than twice as much energy in 1 gram of dried fat than there is 1 gram of dried polysaccharide, starch, or glycogen. If anybody remembers from nutrition, we had carbohydrates or 4 calories per gram, protein, 4 calories net per gram, and then fat was 9. So that's that more than twice as much energy in a given gram of fat. Also, with that density, fat tends to exclude water, where starch kind of draws it in. Think about a potato. Potato is like 98% water. So if you took all the fat in my body, replaced it with an equal amount of carbohydrate on an energy basis, I would be huge. Between the doubling of the carbohydrate and the water associated with that carbohydrate, I'd be even huger than I am already. So we can think about the structure, at least I do. The glycerol molecule is like a dowel or a hanger. And then we have 3 strings of spaghetti hanging off it. Why do I say spaghetti? Energy. There's energy in spaghetti. It's the wrong type of carbohydrate versus fat. But the energy in the triglycerides is in the fatty acids. So more or less inert, relatively speaking, all the energy is down there at the end. So why did I say spaghetti? All the energy is in the tails. So we mentioned already the 3 fatty acids are attached to glycerol by dehydration reaction, but there is no polymer. You don't string triglycerides in long chains. So the elimination of 1 water molecule, got the H from the glycerol, the hydroxide at the acid end of the fatty acid, and that's how those are attached. So we'll stop there. We'll start again at 4 of after your break. Has everybody signed in on the attendance sheet? Has everybody turned in the homework if they have it?
I check to see if I'm on the attendance sheet there.
I think I'd figure it out eventually, but
There you go.
thank you.
Oh, I won't be here next Tuesday.
Okay.
I have a basketball game. We leave in the morning.
So does this mean athletics is open up again?
Yeah, we have a basketball game on Tuesday. We left in the morning.
Okay. Well, I heard over break things got shut down, so.
Yeah, we leave at 10 o'clock on Tuesday morning.
Okay. So should I plan on recording that? Is that where we need to be? Okay. I'll try to remember to do that. What is that, the 15th?
I think so. Look my calendar.
No.
Yeah.
Monday is Single Persons Awareness Day, so I'll assume that Tuesday is the 15th. I assume you won't be back for 240 either, right?
No.
Okay, continuing on. When we're evaluating our triglycerides, it's not uncommon that the chains associated, or the 3 chains that are there, it's not uncommon that they're different. So fatty acids will vary in length. How many carbons are there? And in saturation, how many double bonds do they have, and where are those double bonds located? So I think there's somewhere around 400 different possibilities that you're going to find in milk. DFA put out a thing last year where they were going to analyze your triglycerides, figure out which fatty acids you had, and that could pass judgment on how you're doing in your farm. Are you doing the things you're supposed to, or are the animals under stress? The idea behind that, I don't know if it's a fair thing, and I haven't been waiting for the literature on that, and I haven't seen it. Doesn't mean it isn't there. But if there's less than 16 carbons in the chain, those are usually made in the udder. We call that de novo synthesis, made new. If there's greater than 16, 16 itself is kind of fuzzy. Some are made in the others, some are not. Greater than 16 are generally from outside the udder. They are imported. So they could either come from the fat tissue on the animal's back or from the diet. So the question is, if an animal is doing very well, this should be about 50-50. They are suggesting that if this is much greater than this, something's wrong. So this is 60-40, 70-30. I don't know what numbers they've come up with yet. But if the manufacturer of fatty acids in the udder is shut down for any number of reasons, you might have compromised animals. That to me is pretty fuzzy when you bring things into focus. So I was interested, I asked for the literature based on that. I see where they're going, but I wanted to see how they quantified things. Any questions on triglycerides? Next up is phospholipids. We've got the same sort of structure as triglycerides. We still have that common glycerol. We have 2 fatty acids, but one of the fatty acids is going to be replaced by a phosphate group. So the difference between a triglyceride used for storage of fat and a phospholipid, which is used for structure, is what's hanging off that last carbon in the glycerol molecule. So if we look at that, I think I got you this diagram. So what you're seeing for our phospholipid, we've got our glycerol right here. Off of this N carbon, we got the phosphate group. And then off of this carbon and this carbon, we have 2 fatty acids. So what we have in this phospholipid is a head to it that has charges associated with it. That phosphate group coming in there is going to give that part of the molecule a a polar field, whereas the two fatty acids are still nonpolar, all carbons and hydrogens. So when we see it, usually in a diagram, we see that polar head and the nonpolar tails hanging out. So as we said, you got your phosphate, or not your phosphate, glycerol. You got your phosphate group hanging off. That's got the charges associated with it. And then your two fatty acids, both of which are hydrophobic. There's our symbol. So what happens with our phospholipids, we take that symbol right up here, and you can see that we've got a whole bunch of them that are lined up next to each other with the polar heads facing out toward the water and the hydrophobic tails gathering together. The hydrophobic, afraid of water, they're going to hide behind the hydrophilic heads to get away from water. Has anybody seen this structure before? Biology classes. It's called formally the phospholipid bilayer. So phospholipids, you have this, again, this cartoon, the hydrophilic head, hydrophobic tails. The bilayer, you've got two sheets of phospholipids, bilayer 2, coming together to form our membranes in the body. The phospholipids, very much a structural component of the body. Phospholipids will self-organize if you put them in water. Not a lot of energy to put them together. So the hydrophobic components, those tails, will group together for the same reason that the proteins fold over. The R groups trying to get together, the hydrophobic fatty acid tails trying to get together. So if we take a system like this, the plasma membrane, you've got oil on the top, then we've got colored water, blue water on the bottom. So just like we do with our salad dressing, we shake it up. If we let it sit over time, it will self-organize. All the fat's going to go to the top, cling together. It's going to exclude the water, which is heavier and will fall to the bottom. So what do they organize into? Phospholipids, you put them in water, they form these structures. You might have a situation where all the tails are in the center of a sphere. That's a micelle. You might have a sphere formed with the bilayer. So you've got water on the inside, water on the outside, but you've got a sphere created. And we will get the phospholipid bilayer. So this is the base structure of our cell membrane. We will add to that to get our final membrane. So you got your base structure. We'll add proteins to it, cholesterol, sugars, all kinds of different things to get our final plasma membrane. That might look, if it was a colored cartoon, something like this. So we've changed our cartoon here from blue heads to pink. Tails are still coming off in the middle. This 4-ring structure in here, cholesterol. We've got proteins that span the membrane. We've got sugars that are stuck off the outside that are going to form our final membrane. So phospholipids, our second fat class, are the basic building blocks of all our membranes. So what are the differences between a triglyceride and a phospholipid? How are they different? Give me one way they're different. Okay, so different things there. And what's the other main difference we'll look at? Purpose. Used for energy, used to build things. So build specifically the membranes. So the sameness of the two, what are they same? How are they the same? The glycerol is the same in both cases. And 2 fatty acids hanging off that glycerol is the same. The difference, that one fatty acid traded out for a phosphate group or vice versa, gives it a completely different function. Cholesterol. So how many here have heard bad things about cholesterol? So cholesterol bad, right? Well, actually, it's required from life, so I think we need to be careful. I know I have friends that come from families that heart disease is common. The statin drugs that limit the body's production of cholesterol are probably pretty important to him. But there are other families where it probably doesn't make a difference genetically. So cholesterol, an essential essential molecule to the body, used to make a lot of different things, to help with a lot of different things in the body. Your liver will, as a human, create about 5 grams of cholesterol a day. So guidelines for consumption are less than a 0.5 gram a day. To get up to a 0.5 gram of cholesterol a day, you're going to have to consume a lot of organ meat, brains, livers, things like that. So 10% your intake versus what the body produces. I've never been a big fan of those guidelines. So this is what cholesterol looks like. This is our drawing. Again, remembering that anytime we have a bend in the line, there's an assumption that there's a carbon there with the appropriate hydrogens attached to it. That just makes it easier, cleaner when we're doing our presentation there. So cholesterol has a bad rap, being associated with the plaques, the buildup of the material in the arteries around the heart. A lot of that material is cholesterol. So we assume we have high cholesterol, therefore we have buildup, therefore we run into problems. But I'm not sure that's ever been proven accurately. It might be the other way around. We might have problems and cholesterol ends up there because of the problems. But given that bad rap, there's a number of things in the body that cholesterol is necessary for. So it's an important component in cell membranes. We have a lot of cholesterol. As you can see, that's mostly hydrocarbon from that diagram. They're going to end up inside the phospholipid bilayer. And that helps give the cells some flexibility, helps move those cells around. cholesterol is the main building block of bile salts, which help digest fat. So we take cholesterol, we add some things to it, and it's manufactured in the liver, moved down to the gallbladder. Anytime we have a high-fat meal, those bile salts are excreted into the small intestine to help break up that fat so we can absorb it and use it for our benefit. Cholesterol is also the precursor to all the hormones that are known as steroid hormones. So you've got testosterone, estrogen, progesterone are all are sex hormones. Those are all built in a cascade as one is converted to the other. You'll also have a group of cholesterol-based corticoids that influence the metabolism in the body of glucose, so they're glucocorticoids. You also have a group that influence mineral metabolism in the kidney. What stays, what goes, those are mineralocorticoids. So bile salts, we're adding some stuff here, getting rid of that double bond, changing some things out, making sure that we have some charges associated with that bile salt that are going to help break up, emulsify that fat. So the bile salts, these little sections here, little sections of circles, are going to attach to the bigger glob of fat and break it into smaller droplets. Those droplets, if they've got bile salts all the way around the outside, they become soluble in water, and the enzymes can then get to them, break them apart, so they can be absorbed. Steroid hormones are based on a 4-ring structure. So these dots here are carbon atoms. We didn't draw in the hydrogens in there, just for clarity or less clutter. So you've got 6 carbon rings that are overlapping with 1 5-carbon ring stuck to it. So that's the basic structure of all the steroid hormones. Depending on what's sticking off of that will determine whether it's a form of estrogen, a form of testosterone, a form of progesterone, a mineralocorticoid, or a glucocorticoid. So we start out with cholesterol, and we start systematically adding things and subtracting things to the molecule. So progesterone becomes testosterone, becomes estradiol. These are called the hormone cascades. I won't ask you to remember those, but just understand that one thing leads to another. We take a couple carbons off the top, we get the androgens, where we change the carboxyl group or the carbonyl group, add a hydrogen to it, we take off a carbon, and we're forming our estrogens. So in the gonads, you have this progression. So all of us have all 3 hormones or all 3 types of hormones. It's a question of balance. What's dominant? What's very temporary in the pool that's present? So same thing going through these cascades. They're called steroid cascades, get very, very complicated. have a lot of different steps in them where things are added and subtracted as we move from one to another. This diagram, you have cholesterol starting the cascade. If we go this way, we get our estrogens at the end. If we go this way, we get our mineralocorticoid, our glucocorticoid. But they all have a common overlapping set. Depending on which pathway is activated, which pathway is dominant, will determine what your final products are, the concentrations therein. Finishing up lipids with waxes, usually associated around the skin or like around our ears and nose. We take a fatty acid, we add some more functional groups to it. Alcohol groups, the hydroxyl ions on that fatty acid, and that forms a wax. So waxes, whatever reason, are more hydrophobic than fats, and they make good barriers. They're going to form a barrier that's going to slow down germs from getting through the skin. Slow down the bacteria. If we've got waxes on the outside of our fruits, like an apple, there's a natural wax there. Peaches, all those things have natural waxes on the outside, so they don't dehydrate as well. So if you're going to dehydrate those things, you've got to break that barrier. You've also got insects. Them drying out is not usually a good day. So those waxes are going to slow down that process, allow that water to remain inside of the insect, inside of the fruit. So we'll stop there. We'll start our discussion next time on saturated versus unsaturated and what that means to living animals. Any questions?